and start presenting. Okay, well, welcome everybody to our March 2024 version of Symbiota Support Group. We're really excited to talk to you all today about something that we've kind of touched on before, but this is something that is continuing to evolve in our community, and we're really stoked to chat about it with you. So before we get into the content and discussion, just a couple of announcements. I'll remind everyone that the Digital Data Conference is in Lawrence, Kansas this year at the very end of May. I think it's just after the Memorial Day holiday, so you have a little extra time to get your travel in order. Or, you know, go early and spend Memorial Day in Kansas. I do not know much about what there is to see in Kansas, but I'm sure there's something. And if you applied for one of the biodiversity data fellow travel scholarships or travel grants, those are being, all the applications are being processed right now. We had too many applicants, which was great. That means that there's lots of interest in the conference. And so we're making the selections over the next week or so, and we'll probably be able to get that information to you by mid-March-ish. We also want to let anyone know in the bryophyte and lichen portal worlds that we're going to be having a kind of a combined portal campaign for the bryophyte and lichen consortia. That's going to be upcoming in April and May, and more info will come out via email. So just keep your eyes on your email for those office hours, dates, and all those activities.
Katie, it's Karen here. What does that kind of mean, the campaign?
That's a great question, Karen. The portal advancement campaigns are certain kind of like sprints of time that we set aside to take care of any of the problems, troubleshooting, additional requests that maybe a particular consortium has or a particular portal has. So we focus on data quality, we do a little bit of training, we do a little bit of community development, and we're just trying to get the whole group together and talking and making any changes to their portal that they might need.
Okay, thank you.
Any other announcements from the community or questions about those announcements?
This is Allison. How would I be involved as a small herbarium in the campaign? Would I register? Would I plan to do my projects in those two portals that month? Do I get on an email list for participants?
Yeah, you should probably already be on the list if your contact is affiliated with an institution on the Bryophyte or Lichen Consortium portals. And you'll be receiving an invitation to register for the office hours meetings. And we'll be talking about any of the work that happens in between those, that kind of office hours window.
Thanks.
Thank you. And we still have planned also to have a portal advancement campaign for the Consortium of California Herbaria in August. And I think that there is talk about having a Consortium of Northeastern Herbaria portal advancement campaign sometime probably in the fall. So if you have interest in that, let us know or get in contact with your portal manager. All right, so let's go ahead and jump right in to our topic of the day, which is kind of a revisitation of the extended specimen concept and the extended specimen functionalities in Symbiota portals, what exists and what is coming. So this is actually a little bit of a sneak peek day. So we're going to talk about what we have. I'll give an intro about what extended specimen is. We're going to talk about our new tools, and then we're hoping to get some feedback from y'all about potential new developments in the future and what is important to you. Okay, so as a review, the extended specimen is this concept that has come up in the last few years that talks about the specimen itself as being just kind of one portion of the whole data, the whole usefulness of a record. And that specimen has a digital specimen record. It might have media associated with it. People will call this like the primary extension of the specimen. And then the secondary extension is the fact that you can collect other data about that specimen. So you may have field notes, you may have samples, genetic data, you have field images, you have smaller subsamples or uses of that. And then the tertiary extension is included is data that's kind of related to the specimen because of its taxonomy or because of its locality. So information about the country that it was collected in, information about the taxon that specimen represents. Those can be considered like tertiary extensions. So the idea is that if you just look at one specimen and you imagine all of the types of data that can be linked to that specimen, you actually get this huge web of types of data that can be connected to a specimen. And we're seeing this happen more and more where people have different types of data that they want to link to their specimens. And so Symbiota tools and other collection management systems have been developing lots of ways of accommodating that data. So in Symbiota portals, kind of the main extended data that I'm going to talk about today is that there are tools for supporting traits. There are tools for supporting linked resources. This one's a big extended specimen supporting module. And then there are taxon profiles, which could be considered that tertiary extension. So first, your portal may or may not be enabled with traits. They require someone to actually set them up and to have traits that they want to score. But I know, for example, the Consortium of Northeastern Herbaria and the Consortium of California Herbaria has traits. The National Ecological Observatory Network Biorepository has traits. And I think we're putting together some traits for the lichen and potentially the bryophyte portals as well. So these are things that you can score from the specimen. So they're a trait that applies to the specimen itself, not to the whole taxon necessarily, such as in this case, phenology. So whether it's flowering or fruiting for plants at least. And so for other taxa, for other specimens, it might be something else, like whether a mouse is reproductive or not, or pregnant or not. Or in the future, we would like to be able to put measurements as well. So these traits can be considered categorical, or they could be measurements of that specimen. And the traits are visible in both in the data management interfaces. If I go into a collection that I am managing, there are trait coding tools. Let me zoom in here. So these are ways that you can look at images and code traits from those images. So in some species, it's very easy to know when there's a flower, when there's not, and you can just do that from an image. And there's also the ability to mine traits from textual descriptions. So that maybe the habitat or description or verbatim attributes or dynamic properties fields, you can mine data from there to kind of build up values for traits. And then in some portals, you can also search on those traits. So if you look down here in the Consortium of California Herbaria portal, there are trait criteria available for you to search on right there. So those are traits. Whoops. I guess I just clicked on an example. Here you can see here's a specimen, and there is a traits tab available on the public interface. And the trait data from there goes into a Darwin Core extension called measurement or fact. And so any measurement or trait that has been coded into this specimen record gets put into this separate file. And the reason why it gets put into a separate file is because you can have multiple codings of different traits for one specimen. So you couldn't just put it in the same line of your occurrences spreadsheet. Okay, another big important extended specimen module is the linked resources tab. And I'll pop open a portal again here. If I go into occurrence management and I just go to a random specimen record, and you go to this linked resources. I'm not going to go into detail about all the things you can link, but in general, you can link, let me go back a couple. Associated occurrences. So those are other records that are somehow relationally associated with the current record that you're looking at. So one use for this is often when people collect like mammals, and they will collect all of the ectoparasites off of the mammals. They might put those ectoparasites into another collection of, you know, fleas and mites and whatever they found on those specimens. But then they also deposit that mammal. And so then you can create an association between those two specimens that says, okay, this flea has the host of this particular mammal. And then that mammal is the host of this particular flea. So there's a relationship there. You can also link any specimen to a checklist, to a data set, which isn't listed here. You can link specimens to duplicates. So this is more common in plants and fungi, but you might have collections where someone collects multiple specimens of the same organism or population, and those are considered duplicates, collected on the same day by the same person with the same collector number, and those are linked in a certain way. And then you can also link genetic resources from that page. So I'll bring that back up. Here's where you can put checklist linkages. Here's where you can put duplicate linkages. And here's where you can put genetic linkages. And unfortunately, we don't have any sort of tools that are going to automatically find those linkages. But if you have the information about any sequences that have come out of that specimen, you can put that information here. Okay, I'm going to pause for a second to answer some questions. Karen asks, would this be where we are able to write for bryophytes, CFR for sporophytes, or with GEMA? Are you saying traits, Karen, like the traits tab?
Yeah, because they're like the reproductive things that you could see on a specimen.
Yeah, totally. Yeah, that would be an excellent use of the traits tab. So I think one of the things we're going to focus on in the bryophyte and lichen campaign is to figure out what traits people might want to score on their bryophyte and lichen specimen records and put those in your portals. So stay tuned. For now, you can put that in the reproductive condition field probably, and then you can mine this stuff later to be able to just batch apply the certain trait values to your specimens.
Thank you.
Mary asks, are trait data used in identification keys, or are they a separate thing? Excellent question, Mary. So there's two different things in Symbiota portals. There are traits, and then there are characters. Traits are of a specific specimen record. So a measurement of a specific leaf, the flowering time of a specific specimen. Whereas characters are taxon level traits or characteristics. So that would be something like the flowers are always red, or the tail length is always 3 to 7 centimeters. Those do not currently talk to each other in the Symbiota code. The traits do not influence the character states that you'll see in the identification keys and the glossary. That would be really cool if they did, but they currently do not. Karen has another question. Can you remind me where we should list species co-occurring with a specimen that's contained within a specimen packet? Oh, that is a challenging question. And there might be different answers to this, but I think the currently recommended way to deal with multiple different taxa that are physically present in a packet is to create a new record for each of the taxa in your packet, because each of those is its own occurrence record, its own specimen. And you would want to link those records in your database between one another, and just make sure you put a note in all of those records about where someone could physically find that specimen, because it's in the packet that belongs to X and such species. So of course, if the collection is categorized according to species, they're going to need to walk over to the other species to find that particular record. Oh, Karen, you're muted. Sorry. Oh, sorry. I just muted you.
Okay. But there's only going to be like one like number associated with that specimen.
Yeah, and that's fine. You can have duplicate catalog numbers. You might consider saying like barcode underscore A or underscore B. You might consider adding another catalog number that corresponds to that specific specimen. In vascular plants, and I think in some cryptogams, they will often put multiple barcodes on one specimen. Now, if you have 15 different species in one packet, that gets really onerous really fast. But you might consider how often you have that going on, because some people will use different catalog numbers for all of the species in a packet.
Okay, maybe we'll talk about it during the bride fight.
Sounds good. Yeah, I'm sure that would be a really good community question, because people are dealing with that all the time.
Thank you.
Ryan.
Yeah, so sorry to be contrary, but we definitely don't do that. So we kind of, the barcode or the collector number gets assigned to what the primary reason for what it was collected for, or what it was identified as. And as you probably know, like any lichen specimen is probably going to contain multiple different species on it. So we might put that in like the occurrence remarks, like with species 2, 3, but we're definitely not putting multiple barcodes on one packet.
I won't be doing that either.
So
I would still recommend making a separate record though for those specimens, because if someone is searching for the associated taxon, like say all your data is going to GBIF, like if you're searching for that one taxon, you're not going to find it in the associated taxa field. You're not going to look for it in the associated taxa field. So you're actually losing occurrences of species because they're not searchable.
Well, and so usually what ends up happening is, the rare thing, the unique thing, the less common thing is usually what ends up getting tagged as, what's in the packet.
Mm-hmm.
And the more common things are generally like demoted.
But then you have an underrepresentation of common taxa.
Yeah.
So
yeah,
you still need those.
Yeah, I'm not saying that they're not important, but we have 105,000 packets of lichens at Kahlo, and I can guarantee you just about everyone has got 3 to 10 species in it.
Yeah,
so
for sure.
That would be going from 100,000 to 500,000 occurrence records.
But that sounds more impressive, right?
Yeah. It is. I just think logistically and from a standpoint of having a professional vet the associated information that's on those packets. I mean, that'd be a multi-year grant just for one institution.
For sure. Yeah, some of this is aspirational and like
Yeah.
best practice, but you know, sometimes you just got to do what you can do.
Yeah. So, no, we've had several discussions about that, like, you know, how would you go about not physically splitting specimens, but trying to get all that information in there, and you know, I'm sure at some point somebody's going to be able to do that, but it's not something that we've been able to do or felt comfortable doing.
Yeah. John asks, I'm unsure if this has been discussed in previous meetings, but do y'all have a recommendation on where to include associated iNaturalist observations with a collection record? This is often relevant for my personal collections as I make iNaturalist observations of most of my collections. That's an excellent question, and we do get that a lot. Lindsay, do you remember whether we have developed a documentation on this yet?
I want to say no, but I'll look.
I think it's on your to-do list. The short answer is that people are doing it in lots of different ways. Some people actually include iNaturalist observations as separate records in their portals. I think this is generally not recommended because then you get a bunch of observations and specimens that are the same thing. So what some people are doing is they are linking iNaturalist records as images. They just put a little thumbnail image that says like link to iNaturalist, and then once you click on it, it takes you to the iNaturalist record. But then I think probably a more semantically correct way to do it would be to add it as a linked occurrence. It is a duplicate occurrence, but that's a way that you could put a link to the iNaturalist record in there at least.
Yeah, I just have had trouble even being consistent with myself. You know, I've like there's different options, and I don't I don't see one that really forces me in one way or the other. So I've been trying to I've just been trying to figure out even something just and yeah, you know, there's optional fields. There's plenty of places you can put it, just the link, the notes, but yeah, I don't know where.
Yeah,
the photo thing had occurred to me because that's this winter, we're gonna image our collection, and so those will be associated with each record. But I just don't know.
Yeah. And let's see, Walter's saying that they link iNaturalist URLs into the notes field. Another way you could do it is you could add it as an other catalog number saying like iNaturalist link and then put it
that's a good way.
Like that would show up. Actually, that would show up really nicely. But then when it goes to like GBIF, then it would be listed as another catalog number. So it's not like correct. But Chilla, do you have a comment on this?
I do. I run into this a lot in my work, and I don't have a solution for this. I'm just putting this out there that I love iNaturalist records, and I really would love for them to be linked to the specimens. What happens on GBIF is that if you do that, where you have a herbarium specimen and you have an iNaturalist record, it comes up as separate occurrences because they're pulled from separate portals. Is there any way or is there any plan in the future to make sure to account for that duplicate recording of that occurrence?
Not that I know of, no. I mean, it's kind of the same thing as a botanical duplicate, you know, so, but it is one that is not itself a voucher, but it is an occurrence of a taxon at a place at a time. And so I think duplication is already such a huge, or is just so prevalent that I don't think it's going to go away anytime soon.
Well, no, I mean, I'm hoping, well, actually, I apologize. Some actually might come more from the iNaturalist side because there is a field for collector number on iNaturalist that people do not use, and it's not part of their Darwin 4 export. So it's not being transferred to GBIF because if it had been transferred and people remember to put their collector number on their iNaturalist records, which for the record, I do encourage everybody to do, There is a field for it. And I understand when you're taking an iNaturalist pick in the field, like you might not have your collector number handy, but I do try to go back and write those in. But I'm wondering if I need to like reach out to iNaturalist and be like, this needs to be part of your Darwin Core export, because then you can match up based on the collector number and then, okay, it's a duplicate, done.
Yeah, true.
But just throwing this out there for the whole community, no. They do have a field for collector number, but it is not part of their export to GBIF.
That would be great if they could include that.
So
I foresee an iNaturalist Symbiota Support Group coming, because this seems like a pretty important topic. And then we could walk through all the different ways and have some community discussion about what we think is the best way.
I love that.
Some like usable ways. Cool, great. Thanks for all your questions and ideas. Let me see if I missed anything else. Allison said, when determiners note the gemme, I've been putting them in determination notes. Sounds like they should also go in the traits field for searchability. Yeah, I don't think that you currently have the ability to add those as traits, Allison, or to code them as traits, but that could be something that happens in the future for sure. Let's see. Diego notes, if a record has more than one other catalog number associated with it, but no catalog number or barcode, is there a way to select which other catalog number to map the data to? For example, by using the tag associated with the other catalog number. Are you saying like when you're doing an upload, or when you're just trying to put like an iNaturalist link in there, or?
Yes, this is during an upload.
Oh, during an upload. No, there is not a way to do that. It's just going to search for any other catalog number value that matches that record.
Okay, thanks.
Okay, and there is some more good discussion about multiple specimens with multiple taxa in the chat, so I recommend taking a look at that. All right, I'm going to pop back to my presentation. Okay, the last extension I wanted to talk about briefly is taxon profiles. And so when you go and search in a specimen in a portal, let's just say I'm going to do a search for this Helianthus annuus. Annuus. If you click on one of the taxon names of a specimen, rather than taking you to the specimen itself, if you wanted to do that, you would go to full record details. But rather than taking you to the specimen, it takes you to the taxon profile page. And so this is information about that taxon, not necessarily that specimen. There are any descriptions that are added, links to any resources that might exist. And this can also give you a place to search for all the occurrences of that taxon, to look in the taxonomic tree for where that falls in the taxonomic thesaurus of this particular portal. And then, yeah, other external resources and taxonomic resources as have been applied here. And another thing, if traits are available in the portal, this is also where you can look at kind of the aggregated trait values for that taxon. At least for phenological traits, there are these really cool graphs that show you values for all the specimens in this portal that have trait data. This is what the trait data are aggregated. So it flowers mostly in these months, or at least that's when most flowering specimens have been collected. So that's pretty neat as well. And that does come from the portal data. We talked about checklists and data sets very briefly, but hopefully you guys are all familiar with checklists. And if you are not, Lindsay can throw a link in the chat about how to get more information about checklists. Super useful tool. Okay, so here's the sneak peek part where we talk about new, oh wait, Karen has a question. Maybe those graphs reflect when people are collecting. Yeah, you definitely have to interpret the data with the biases of collecting activity in mind. You would like best compare the non-flowering graph with the flowering graph and see if they are kind of overlapping or not. Okay, so new tools in the works. One of them, I think this is going to be in high demand, is a batch linked resources uploader. So that linked resources tab that I showed you a few slides ago, that you can currently only add one associated occurrence at a time. And that can get very onerous, especially if you have like a lot of subsamples that you want to upload. Well, thanks to a grant or a contract with the Fort Worth Botanical Garden and Botanical Research Institute of Texas, George Mason University, and the California Botanic Garden, we are developing a tool that will enable us to batch upload those associations. So soon you're going to be able to extend your specimens really fast, and that's very exciting for us all to have. There's also going to be a tool for batch tagging images. And this is not necessarily by uploading something, but there's going to be an interface where you can look at images and apply certain tags to them if you have permissions to edit those specimen records. So that's thanks in part to the Eastern Seaboard TCN and also the Big B TCN or Thematic Collections Networks. Those grants enabled us to develop these tools. And then we're also making improvements to the material sample module. You may have heard about the material sample module. It's only kind of in use here and there. I don't know if there's been an official rollout of it because it's still being improved upon slowly and iteratively. And that's mostly thanks to the National Ecological Observatory Network funding. So we're really excited to kind of roll those out more to the public. We have some documentation already on Symbiota Docs about what material samples do and how you can use them. So feel free to check those out, and Lindsay will put that link in the chat. And that just reminds me to emphasize that remember that development, like actually making new stuff in Symbiota, is not supported by our current IDBio Symbiota Support Hub funding. Like that's pretty much just for maintaining the portals that we have and making sure that they're functioning and that they're up to date and that we can answer all of your questions and lead these webinars. But the actual coding stuff gets developed because of grants and contracts with other folks. So if you have anything that you're just dying to see in the portals, consider putting that into a grant application, or if you have other sources of funding, that would be a good thing to chat with us about how you can get that on the development priorities. John asks, would this batch image tool be for images of herbarium sheets? It's just for whatever images you have in your portal. So if you're doing it for herbarium specimens, then you could batch tag herbarium sheets. But I think it was originally developed mostly for bee images because they had different orientations of the bees, and some of them had labels and some of them didn't. So that's being developed to help batch tag those. All right, let's go into community priorities for future development. So this is where Lindsay combed through all of our wish list items, and we brought out some of the main kind of extended specimen related things that we thought the community was vying for. And so we want to make sure that our thoughts on this or our understanding of the demand is correct. So a few of these community priorities that were outlined are a references module. So this would be an ability to link literature or permits related to your specimens to those specimen records. We think that that's in pretty high demand. The ability to search for material samples and their multiple properties. I know that this one actually is in development, so this one is happening, but we still want to see how important it is to folks. The ability to add PDFs to linked resources. So again, maybe uploading a permit or some other documentation relating to a specimen. The ability to index agents, and by agent, I mean like a person who is doing something relating to a specimen. So that might be like a preparator, a collector, or an identifier. So being able to link unique identifiers relating to those people to their names. And so this would be able to deduplicate names. You know, if you have Juan Hernandez, J Hernandez, J.M. Hernandez, you know, all of the versions of a name, you could link them all together with a unique identifier, and then also be able to link them to their Bionomia profile, if you know what that's about. We've seen a demand for improved commenting functionalities. So like the ability for people to leave contact information or for admins to receive notification emails when there is a comment or some other interface for being able to review comments. And then we know that people are really wanting to score and store numeric trait data. So like measurements and such. Okay, so this is where we start the discussion. And I think number one in the discussion is we just want to get a poll of all of y'all. And so I just submitted opened a poll for you to answer which of the ones mentioned here are your highest priorities. And I think that'll just be interesting for us to all see right here in this meeting. So go ahead and give those a think. And then we can talk about the results. And also if there are other things that you're like, whoa, this is a huge priority for us, but this isn't mentioned in your list. So we'd love to hear those answers as well. So I'm going to leave this open for 2 or 3 minutes and we'll collect your answers. And if you need a reminder about what any of the poll options mean, just ask. Yeah, Chandra, I can put that up. Give me a sec.
I think I prematurely ended the poll, Katie.
Oh no, here we go. Can I relaunch? Oh no. I'm sorry, everyone. You're gonna have to re-enter your poll.
My bad, everybody.
It's okay. It's all right. I'm gonna bring open the extended specimen graph while we're thinking about this, just to give you any ideas. Thanks for that recommendation, Chandra. So if you'll notice, the first question is just like select all the ones that you're interested in. The second question is like select your highest priority. And then the last question is if you had any other ideas that we haven't actually listed here. Right, we're seeing some pretty good participation. Nice. Oh, and we have 8 entries for others, so I'm excited to see what those are. 9 entries for other.
You can't be surprised by that. Herd of cats we are.
No, I'm actually surprised that it's not 23. All right, I'm going to close the poll in 5, 4, 3, 2, 1. Okie dokie. Okay, very fun. I think I can share my results now here. I'm not exactly sure what you can see, but it looks like everyone is interested in everything. So that's fun. Okay, not everyone. 3/4 of y'all are interested in the references module, a lot of PDF linkages. And then the highest priority for the majority of people appears to be agent indexing. So adding people identifiers to the people associated with your collections. So that's cool.
Are you sharing your screen? We're not seeing anything on our end.
It says that I'm sharing the poll, but I don't think it's sharing the screen.
Okay. Oh, I can see it. I wasn't sure if only I could see it because
no, I think everyone should be able to look at the
Okay.
poll results now. Okay, now let's see if I can find. Okay, in a pop-out window. Thanks for that, Dakota. Now I want to see your short answers. I might have to do some signing in here. All right, is anyone surprised by these results? Anything to note? Okay, here we go. Here are the free text responses. Ability to link specimens across portals. Oh, that already exists in the linked resources tab. You can do that. Indexed and searchable preparations field. Cool. Link reference to identification source. I guess you can kind of do that in identification references, the field, but it's not, indexed or anything. Customizable label output for printing labels, which does exist, but it doesn't exist in a Microsoft Office Word file. Characteristics of fruiting. So I think those would be things that you put in the traits. So I think that exists. We would just have to make traits in your portal for it to be useful for you. Links to audio and video. Yeah, very important. Different types of media. Biocultural and traditional knowledge labels. multiple points of contact per collection for comments. Oh, interesting. That, yeah. So like when you look at a public display of your specimen record, I think only one is listed as the like email this person for more info. And then field images. So you can put field images along with your specimens. They would just be, you would have to label them according to the catalog number if you're batch uploading them. But if you're not, you can just upload them one by one into the portal. So you still could put those associated with your specimen records. Mary asks, how can we create an ID sources reference table? So are you saying like,
well, you said we could do the reference stuff. I hate writing out Flora Pakistan, you know, each time. It's a lot to write out on each specimen.
Mm-hmm. Like you mean being able to select from a drop down or what?
Yeah, something like that.
Gotcha. Okay. I see. So like being able to select, I got this from Flora of North America or Flora of Pakistan or something like that.
Because it's something I think a lot of us ignore. Heck, I never did it when I was young. And there's going to be times when it won't fit, but sometimes it does.
Mm-hmm. Yeah, it is important to note whose idea of a certain taxon are you aligning with when you're putting this.
And when did you do it? Because they change sometimes.
Yeah, for sure. Well, thanks for that feedback, everybody. Did any of the free text responses strike a chord with anyone? Or do you have any other comments about the ones that we mentioned? Or maybe you remembered something that you forgot to put in the poll? Interested in all responses.
Yeah, I put it in the chat. Mounted date. I don't know if there's an idea of adding that into or adding that as a field, because I think at USEH in Clemson as well, I edit multiple data sets. We basically put that in the data generalizations because we don't know where else to place it.
Mm-hmm.
But if there is a better place to put it, I would love to hear from everybody. Because I wasn't 100% sure. So I think, yeah, basically anything that doesn't fit anywhere else, we put into data generalization so that we can search it.
Mm-hmm.
But that is one that we use heavily at USCH.
Mm-hmm. Yeah, I wonder, I think because Symbiota is so Darwin core focused, there are a lot of like curatorial type fields that are not included because they're not part of the Darwin core. I wonder if there's a prepared date though. I don't remember.
There's it's in material sample. So if like that's not enabled, you wouldn't be able to see it.
Gotcha. Oh, Ryan says georeference date. Yeah, that is a Darwin core term that we just straight up don't have. Karen says some improved label making options. We have a really customizable label making tool. Maybe we should have a Symbiota support hub on that.
That sounds good. I'm still struggling to get it.
It's a bit complicated putting it together, but once you got it locked in, it can be really good.
Thanks.
Label making support. Okay, it sounds like we're getting a lot of support for that. So that's great. We have 2 more Symbiota Support Group topics already lined up. Thanks, Lindsay. All right. Are there any other topics, questions, thoughts? They don't have to necessarily be extended specimen related.
I did have one. Go ahead.
I was just going to ask you to explain the search materials samples a little bit more. I don't really understand what that is.
Yeah, so we'll probably have a more extended discussion about material samples in the future, but material samples in general it's a module for you to put any subsamples of a particular or of your records. And it's associated with that specimen rather than creating a brand new occurrence record that is also in your collection. You instead are making what's called a material sample occurrence. And this is being used pretty heavily in the National Ecological Observatory Network because they're taking like tissue samples and DNA samples and all sorts of other things. And they're linking those as material samples. And so they're hoping to be able to search for those things or search for specimens that, like, hey, this one has already been sampled for DNA. So I want to make sure that I'm requesting a loan of all of those tissues rather than trying to do destructive sampling of new specimens, for example. Chilla, you had a comment or question?
Yeah, when I download information from the determination history table, the Cernak taxon ID doesn't come with it. Is that on purpose? Is that something that is going to be fixed in the future, or what's the thought process behind that?
I think that probably is on purpose because Symbiota portals were not designed to be taxonomic resources. And so I think the thought is that taxon IDs applied to taxa in a Symbiota portal shouldn't be considered like permanent links or unique identifiers. Instead, that should be coming from some other more authoritative taxonomic resource.
Because in the occurrence data, there's a Cernak taxon ID, but there's not one in the determination history table.
Mm-hmm.
Okay. Well, the reason why I asked about that is because I've been working on a synonymy table specifically for South Carolina, and then I was surprised. I used the Cernak tax ID to set that up. And then I was surprised when it wasn't in the determination history.
That could just be a bug. Like maybe we should just put that on the bug list.
Well, because I wouldn't see why. Yeah, am I just me misunderstanding? If that is something that you guys could add since it's already part of the occurrence data, if that's just like an extra thing to add to the export of the determination history, I would really appreciate that. But I also understand where you guys are coming from.
Is that are you saying that the field exists but it's all null, or that it's not a field that exists?
It's not a field that exists.
Gotcha.
So like
I'm wondering whether that is considered a field in the determinations extension, because we get our file type from the Darwin Core, but I'm sure Darwin Core has a field for an ID of your taxon in the determinations table. So
right. Well, now I'm questioning myself. Let me double check. I'm pretty sure the last time I did it, I was surprised. I was like, yeah, because I click include determination history, and then you download the data. And then here, let me just open it up, just make sure I'm not insane. Let's see. Yeah, it does not have Cernak taxon ID.
Interesting. Yeah, that seems like something we should probably harmonize.
Thank you.
Other questions, comments?
Katie, I have just a quick question. We're starting a sort of a long-term project to collect seed material. We started building like a little seed repository, and we were planning on keeping accession seed material, but also, some of it would go into cultivation, stuff like that. So I'm just guess the general question, I think I could start figuring out specifics, but is Symbiota portals like with the Cernak portal be an appropriate place to start building the seed collection, or should I look? I just kind of want to. Is that a good place to start?
I think we have some budding, pun not intended, seed collections out there. There's not like a dedicated solution yet, but I know that a lot of people are interested in looking into this. Like, for example, Botanical Research Institute of Texas, I think they also working on seeds.
Yeah, we collaborate with them.
Yeah. So I think it is a possible solution. It just hasn't really been thoroughly explored yet.
I was thinking about setting up a separate collection, basically a new herbaria, but it'd be focused on this seed collection that we're building. Seems like
I know that has been done in some collections. Some might consider that would be a good use of material sample.
I was wondering about that too. Yeah.
But that depends on whether there's like a specimen and then a bunch of seeds taken from it, or like a voucher specimen for the population. And
yeah, maybe not always. They might not all different combinations will occur.
It's definitely worth some more exploring for sure.
Thanks.
Any other comments, questions? All right, well, thanks for your time, everybody, and for your feedback. That was very useful information to gather. I have it all saved in my Zoom polls. And thanks also for the ideas for the future Symbiota support groups. Those are going to be great topics to cover. And we look forward to chatting with you more. And as always, feel free to send any follow-up questions or thoughts to us at help@symbiota.org. And we're happy to gather your feedback there too. Thanks, everyone.
Thank you.